which is okay. Feline, could you try to turn the music down a little bit? I can't think. I'm in summer mode. I can't multitask as well as I should. So what I want to talk today about is, I know a lot of you have your ideas. Not all of you have ideas for doing research, but a lot of times what I've seen over my time as a teacher and helping students do research is we get so excited about our research topics, we just want to start the research before we really think about everything that needs to be planned ahead of time. And so what I want to do today, I'm not telling you, I'm not going to be telling you anything very specific for your research projects, but I want to give you an overview of what I think you should be thinking about. Hopefully I at least mentioned one or two things that you haven't really thought about that you can think about now before you do your research. This summer, we've been trying to help a lot of you just kind of get into the lab and do something because we wanted you to do hands-on projects. But when you actually sit down to do your formal research project, there's a lot of planning that needs to go in ahead of time. And so that's kind of what we're talking about here today. All right, you've done some really cool stuff this summer, but once you sit down and turn this into like a science fair project, what do you actually have to do? So we're going to talk about how to do that. I'm guessing no one's ever heard of this guy, John Tukey. I'd be very surprised. Brandon. So I just like this quote. This guy was not a scientist, he was a mathematician. He did math for a living, right? But he's famous for this quote: finding the right question is often more important than finding the right answer. And that is so true of science. The hardest thing for us to help you guys with, as teachers helping students or scientists helping young scientists, is what are the important questions worth asking? So hopefully you've had time to think about that this summer a little bit. And so the reason why I put this quote up here is picking a research topic shouldn't be about doing a skill that's really cool. Like you saw us doing something in the lab. That's not really a good way to pick a research project. The best way to pick a research project is to see something really interesting and ask a question about it. So Serena, I'm going to pick on you a little bit because you told me you have an idea. What's your research topic?
Well, I'm still thinking about it, but right now it's recovering plants.
For what?
Recovering plants, but I have it.
Like how to get plants to heal?
I'm using hormones.
Okay, very cool. So do you have, that question is like how to get plants to grow better, heal better? All right, cool. Anyone else have a pretty strong idea what you think you're going to be working on for the rest of the school year, or at least a research project? Elena?
I'm taking the juices out of vegetables and starting to do stellium cells, so they mimic cancer cells, and then testing the juices on them to see how they react.
So will different vegetables from juices help heal these cells or get their cells to grow better? Right? So boom, there already, you have your question already, which is really cool for both of you. And if the rest of you aren't there yet, that's okay. But these are some of the things you want to think about before you get to your project. Anyone else have a pretty firm research idea? Enrique?
I guess
let me go with Justin first, and then I'll come back to you. Go ahead, Justin.
I'm gonna have a cancer cell from a specific body and treat them different diet to see how they grow.
Okay, so similar thing, but adjusting the food or adjusting the nutrition. All right, very cool. Enrique?
I guess light refraction.
So light refraction, I know a little bit. So Enrique is interested in making a hologram. So, right? Light reflection, holograms. So we've been working on that a little bit. So what's the question then? That's what we've started with. Okay, so finding the best medium to make your hologram or whatever? Maybe, okay. Maybe I'm putting words in your mouth. All right. So anyway, I'll get started here so I don't keep you here too long. I want this to be kind of interactive. So if you have questions or if you, something pops in your head, just raise your hand or shout it out. That's good thing. So my first question to you is, what is experimental design? So when we talk about doing an experiment, what do we mean?
Like to figure out something that someone wants to know.
Okay, so you're answering a question, you're figuring out something that you want to know. Good. Anything else? That's a great start. Thank you. Okay. All right. So this is a textbook definition. So this is like school. I know we told you this summer was going to be like school, but this is going to be a little bit like school. I don't like this definition, like the whole thing, but I like parts of it. So here's what a textbook would say experimental design is. The purposeful integration of research objectives. All right, so I'm going to stop right there. We're going to combine some things. Right? We're going to combine our research objective. What is our question or what is our goal? Right? We want to heal Dicty cells. We want to improve plant growth. We want to create a holographic projector. Right? That's a research objective. And in order to do that, what do we need to do? Okay, so in order to answer a question scientifically, we need to collect data. Right? We need to analyze that data. And we need to make sure that we have a lot of data, quantity, and that data is good. And I told you this is like school again, so just bear with me. But really, so what this definition says is, if you want to answer your research question, you need to figure out how to collect your data. You need to collect your data. You need to analyze it. And then the last part that's on here is really, we need to share that information. So that's kind of what this whole PowerPoint presentation is going to be about. How do you start with an idea? How do you start with a question and then physically do your experiment? This is the fun part. I know, like, not me talking about it. Doing the experiment should be the fun part. The tough part is what you've been doing this summer. Thinking about the ideas. What are the questions worth asking, right? That's the part where there's just so many options. But the fun part for me, like doing experiments, is kind of like doing a puzzle. Because there's so many ways that you can do it. There's so many things that could go wrong. You have to problem through trial and error, figure out the right way to do it, which I think is good. Okay, questions on this definition? Anything else pop into your heads while I was talking? Okay. All right, I'm going to skim over this. Okay, I'm going to start here. So to me, especially for you guys, most of you probably doing your first experiment or maybe the first couple years of doing experiments, there are really two different kinds of studies that you can do. Some of you, if you decide to do a research project, like a science fair project, will do what we call observational studies. These definitions are these are more geared for my high school kids, so if they don't make sense, I'm going to try to simplify them by saying, but if these things don't make sense, just ask or let me know. So, to do an observational study, you really don't set anything up in a lab. So there are some studies, especially studies for like the environment, where you don't get to control things. You just watch what happens and then make observations about it. The best example, now again, these examples are maybe a little complex, but hopefully they'll make sense. So imagine you're doing, you're like a medical doctor, and you want to do a study to see how someone's diet, the foods that we eat, so Justin, maybe kind of like yours, how the foods that people eat affect something about their health, like blood sugar levels. So if you're a scientist and you're doing an observational study, you wouldn't get people and force them to eat food and then measure their blood sugar levels. What would you do? What would be an observational study based on this?
Get people that already do that diet.
Yeah, get people that are already eating those different diets and then just compare, right? You have to collect some data, but you're not controlling the experiment. That makes sense? Another kind of study that I think would be really cool, that would be more of an observational study. We had a couple of people come in and talk about mosquitoes this year. I'm really interested in what kind of mosquito species are different in cities versus rural areas. I live in Wobblesdorf, like pretty rural area, right? But I wonder if the mosquitoes that we have at my house are different from mosquitoes in Robisonia or in Reading or in Philadelphia. So if you wanted to do an observational study like that, you could collect mosquitoes out in farm fields, collect mosquitoes in a city, and compare them. You're not designing the experiment. You're not controlling anything in the experiment. You're just looking at the results. Does that make sense? Okay. And then you guys are smart, so of course that makes sense. All right. The other kind of study that you could do is an experimental study, right? In an experimental study, again, things are maybe a little bit complex, but you are manipulating something. You are physically changing something about the experiment. And so if we take the same example about like diet and blood sugar levels, maybe in this example, you might physically, like patients could be randomly assigned to different groups and we feed them different diets. Could we do that with people? Yeah, well, physically we could. We couldn't do it here at the high school or middle school level. Could you do an experimental study with fruit flies? Yeah. Could you take fruit flies and put them into 2 different tubes and feed them different things and see how it affects whatever? Yeah, absolutely, right? So, in any case, I just wanted to make the point that some of you will probably end up doing observational studies, where, like, especially if you ask people a survey, maybe like at Readingbrook Science and Engineering Fair, you can design surveys, send it out to people, get questions, and that can be a research project. That would be an observational study. That's totally fine. Many of you probably want to actually build something or design something. That's an experimental study, and that's what it is. Okay, thoughts, questions? Am I boring you? I think this is exciting. I know I'm a geek though, so that's okay. All right. Okay, this is where it's gonna get, so you've probably heard a lecture kind of like this before in school, in like one of your science classes. I'm gonna talk about things for experiments like control group and experimental group, or independent variable and dependent variable. Do those things sound kind of familiar? A little bit? I've been learning about this stuff for a long time and I still need a reminder every once in a while. So if you feel like you've learned this but you can't quite remember what some of these terms mean, that's okay. All right, so imagine the most simple experiment you could possibly imagine. For me as a biology guy, that would be an experiment like, I want to take 2 plants. I'm going to put them in the same soil, I'm going to put them in a pot, I'm going to put them in the same, kind of like the same part of a brew, like in a window. But to one of those plants, I'm going to add fertilizer, and to one of those, I'm not going to. Right? So, the simplest possible experiment that you can imagine. In that experiment, or in any other really complicated experiment, we always have to be comparing. Right? And so, every experiment, even the simplest experiments, have something that we call treatment groups. You'll hear other people call it, like, give that maybe a different word, but typically every experiment that we do has to have something called a control group and an experimental group, or a control treatment and an experimental treatment. Do those words sound familiar to everybody? Control and experimental? Okay, so what's the difference between the two? Matai?
A control group is a group of organisms that you keep for reference, and an experimental group is a group that you try to experimentally change.
Could you say that a little bit louder? I'm sorry.
A control group is a group of organisms that you use for reference, while an experimental group is an organism that you change. For example, if I wanted to change the genetics in one organism, I would change my experimental group, but keep the control group the same.
Okay, perfect. Yeah, that's a great answer. The one thing I would add to that, it doesn't have to be living things, right? A control group could be like in a computer program. A control group could be, I know you guys are more interested in like the VR stuff. Like control group is basically what you are not, what you're not changing. So in any experiment, no matter whether it's, I'm picking on you guys back here now because I know you're interested in like the VR and computers, 3D printing, right?
Yeah.
Whether you do a project like that, or you do a project with cells and plants, or holographic displays, whatever it might be, in every experiment we need to have a control group and experimental group. And the most important thing that I can stress to you guys is those 2 groups need to be exactly the same. Exactly the same. Except for the one thing that you're interested in studying. Except for the one question that you're asking about those 2. So if it's a computer code or a computer program, or if we're going to 3D print something, we're going to 3D print these 2 things exactly the same. Except we're only going to change, I don't know, what can you change on a 3D printer?
Size.
The size. The temperature that we're going to extrude at. The plastic film, we're going to use 2 different plastic filaments for the 2. Okay? Or for Justin, right? We're going to, everything can be exactly the same, but the diet is going to be slightly different. Or Enrique, everything's going to be the same except the plastic that we project onto. Does that make sense? Okay, sweet. So the control group is typically the one we say is like, we leave it the same, we're not changing much. The experimental group is where we add that thing in that we're interested in studying. Okay. Moving right through. So we have our research question. That's the first thing we have to do. The next thing is kind of think about what are the groups going to be in our experiment. The next thing, and probably the trickiest part of any experiment, is working with our variables. So what's a variable in general? Math class, science class?
Something that can change something.
Yeah, something that changes, right? So in our experiments, we're talking about experimental design. In an experiment, something that can change. And the reason why I think that's tricky is because when we do experiments, some variables are good. I'm going to use quotes because I don't know if anything's really good or bad, but some variables we want to change, other variables we don't want to change. So experiments are all about keeping as many things as like the same as we can and then just changing a few things. So again, textbooky words, the parameters that define an experiment, but really anything that can change in an experiment. There are 3 different types of variables that you have to think about and know about and write down in your lab notebook before you start your experiment. And I remember hating this in high school and in middle school and in college because my professors and teachers always asked, whenever we would do a lab, what's your independent variable? I don't know. Right? But you have to know. It's really important. So looking back, I know why they were asking me to say those things. Because you can't do a good experiment unless you know what all your variables are. So independent variable, dependent variable, and then the last one is, I call it an extraneous variable, but there's other words. So I don't, do most of you, independent and dependent variables sound most familiar? Did any of you learn those as experimental variables and response variables? All right, so independent variable. What is the independent variable? I know the definition's there, but I want to hear it in someone's words.
A part of matter that is directly manifest.
What is your definition? Don't read it off the board. But that is a correct definition, I hope. I guess this part will be more of I'll try to block you guys. Usually I can stand in front of the board so you can't see. Let me do this. What's an independent variable? Okay.
Variable is like basically something that depends on the other so it can basically give.
I would say that's more like the dependent variable. It'll be easier maybe to define when we get there. Okay, it's really, I would say independent variable, I was always confused about this when I was in school. It's taken me a long time for it to sink into my brain. I'm slow when it comes to that. The independent variable is what you purposely change in the experiment. That's it, right? Don't worry about all this other stuff. The independent variable is what you change. Right, so Enrique, if we're going to try this on water vapor and carbon dioxide and plastic, right? That's your independent variable. That's what you would be changing. So Elena, what would be your independent variable? What are you changing on purpose?
The different juices.
Right, exactly. The juices extracted from different vegetables. Awesome. Anyone else have there? Their research ideas sorted out? You did one last year with your filters, right? So last year, she was interested in filtering, was it salt water? Filtering salt water with different kinds of really tiny micro filters. So your independent variable is just a type of filter, right? So yeah?
Instead of watering like the plants with water, I'm doing hormones. Water.
Exactly. So which hormone are you adding, right? That would be your independent variable. Good. Okay, so independent variable is pretty simple. What are you purposely changing in your experiment? You can't start an experiment until you know what your independent variables are. That makes sense, right? Okay. All right, dependent variable. Then, and Enrique, I think this is a little bit more because it depends on what you change. The dependent variable is really the data that you collect. A scientist would say the dependent variable is the parameter that changes as a result of the experiment. But really it's what are you looking for at the end of your experiment? So Elena, I'll pick on you again. So if you change your vegetable juices, what's the data that you're going to be collecting?
How much they affect the Dickley cells.
Yeah, so maybe it's the survival rate of the Dickley cells, maybe it's the growth rate, maybe you're looking to see if they're like infected or not, I don't know, right? So is there just one dependent variable that you might be measuring? Not necessarily. You can measure 5 or 6 things about those cells, but that all kind of falls under dependent variable. It's the data that you're collecting, it's the result of you doing your experiment. So Serena, what are you planning to look for? Not sure yet. So that's good. You haven't started your experiment yet, right? And again, sometimes when we're doing experiments, things pop up. Oh, I wish I would have collected that data, right? Is that what happened to you last year? Yeah, like if we don't plan well enough, we don't think about all the possibilities. We get to the end of an experiment thinking, I just spent like a month doing that experiment, or I just spent a whole weekend doing that experiment, and I wish I would have collected that. That happens sometimes. You're never gonna totally avoid that, but if you plan a lot ahead of time, it helps with that.
It could be for it to grow, the plant to grow.
So like growth rate, number of leaves, do they produce, depends on the plant, do they produce flowers or not? I love to garden, so I'm always interested, like, if I add fertilizer, is that just gonna make the plants grow taller, or am I gonna get like more tomatoes or more cucumbers or whatever, right? So, interesting question. Okay. Any other thoughts, questions, independent and dependent variable? Okay. The last type of variable, some people call them extraneous, some people call them confounding. Both of these words, these are the extra variables that we need to control. Confounding variables are, if we don't consider them, they might confuse the results of our experiment. But in any case, these are things that interfere with our experiment. So between the control group control group and experimental group, what do we need to make sure happens with these variables? So if we look at, maybe Serena, I'll pick on you because the plants are easy. So you're going to take the same plant, put them in the same soil, give them so you're going to have your 2 groups, right? Everything's going to be the same. You're going to add hormones to some and not hormones to some. Those are your control and experimental groups. What else needs to stay the same about those 2 different groups? Anyone can answer. It doesn't just have to be her.
Everything else has to stay the same.
What's that?
Everything else.
Right, be specific. So when we're working with plants, what do we need to make sure stays the same?
Soil.
The type of soil.
How much you're putting like the watering and hormones.
Yeah, how much water we're adding.
Type of plant.
The type of plant, absolutely. The sunlight, the humidity, is there breeze or not, right? Now, is it possible to control everything? No. But we have to try. And that's why we do experiments in laboratories, because we can control those kinds of things. If you would do an experiment at your house, it might be really difficult to control temperatures, or control the amount of sunlight, or make sure that the temperature doesn't dip down at night or go up during the day, whatever it might be. But in a laboratory, we can do that, especially when we're working with things like bacteria. Matthi's been growing bacteria most of the summer here at SRI. We've kept them at 37 degrees Celsius the whole time, right, Matthi? Or that'd be difficult to do at home. 37 degrees is our body temperature, 98.6 degrees Fahrenheit. So extraneous variables are all of the things that we need to try to control. Okay, I always use this example. So I'd like you to just think. I'm going to make you think for like a minute. Here's a scenario. I want you to just identify what you think is the independent variable, the dependent variable, and the extraneous variable. You can work with the person next to you. Okay. So here's the example. A herpetologist. What the heck's a herpetologist? You know that?
A person who studies reptiles?
Yeah, a person who studies reptiles and amphibians. So a herpetologist wants to find out if endangered sea turtles produce the same number of eggs in sandy beaches versus beaches that have rocks or pebbles. So if we would turn that scenario into an experiment, what would be the independent variable, the dependent variable, and then Nate, I'd like you to just think about 1 or 2 extraneous variables that we'd also like to kind of control for. All right, so I'll give you like 30 seconds. there. Independent, dependent. I should sit on the computer. All right, anyone have an idea? For independent variable.
The energy variable would be like the different types of beaches.
Yeah, absolutely, good. So in this case, independent variable, the thing that the researcher is purposely changing is the type of the beach, right? Is it a sandy beach? Is it a pebble beach? Awesome. What about dependent variable?
Type of sea turtle. Type of sea turtle.
Oh, hold that thought. I don't think that's the dependent variable, and I'll explain why in a minute. So in general, let's define dependent variable. What is the dependent variable again? Not for this scenario, but in general. So what?
Variable that depends on the variable that changes.
The variable that changes because of what we did, right? So, or the data that we collect. So what, in this case, what data is the researcher trying to collect? Yeah, exactly, the number of eggs, right? So, that's what you're gonna say. Okay, so, let's say they count and there's like 16 eggs in the sandy, I don't know, do you get it? So sea turtles go up on the beaches, they dig a hole, lay their eggs, cover them up, and then say peace out and never see their babies again. It's an interesting strategy. But anyway, so maybe there's like 16 in the sandy beaches and there's only like 12 in the beaches with rocks. So yeah, the dependent variable will be the number of eggs. Extraneous variables, things that the researcher would want to make sure is the same between those groups. One of them would be the species of sea turtle. Yeah, it could be like a loggerhead sea turtle or a, it's the only sea turtle I know. What other kinds of sea turtles are there? Ridley sea turtle? Right. So I don't know a ton about sea turtles, but we need to make sure that, because maybe loggerhead sea turtles normally lay 15 or so eggs, but Ridley sea turtles only lay 12, I don't know, 10 eggs per batch or whatever. So, okay, good. What else would need to remain the same? This would be hard to, this would be a really hard experiment to control. This would be more of an observational one, but.
The weather.
Yeah, like the weather. Maybe, like, is the water temperature, or is it like stormy where the sandy beaches are versus the rocky beaches are, right? They would want the exact same weather. Good. What else? One or two more.
Predators that live there.
Yeah, you'd want to make sure that there either were no predators or there were the same predators in those two beaches, right? And then there was what eats sea turtle eggs? Gulls, birds. Okay, all right. Make sense for everybody? All right, you guys are All right, let's skip this part. Okay, last couple things. I want to talk a little bit about hypothesis. I know you've learned about them, but I'll just go over that quickly. But in general, this is kind of like a side note. When you finally do your experiment, whether it's here at Common Wiser, or back at your home school, or at home, wherever you decide to do your experiment, I beg you, please, before you start, think about collecting as much data as you possibly can. Collect lots of data. Because if you don't, once you've done your experiment, depending on what your experiment's all about, it might be really difficult to go back and restart and collect more data. So before you start, you need to make sure that you've really thought about, especially with something like plants or something like a living thing, like once the plant grows, or once and then dies, you can't measure number of leaves anymore. You can't measure the growth rate. Some things are just difficult once your experiment is already started. So it's better to have more data than you need than too little. One example, we had a really interesting project a couple years ago. There was a student that was interested in celiac disease. You guys know what celiac disease is? Celiac disease is when people are allergic to an ingredient in wheat. And so they have bad allergic reactions to things like pasta or bread, pizza dough. Yeah, it sounds terrible. Right? Those are all the things that I love to eat. But what he wanted to do is he wanted to see if he could detect celiac disease by taking people's saliva. Right now, in order to diagnose celiac disease, you have to draw someone's blood. She said, well, it'd be a lot easier and less painful if you could just do it from saliva. So he went around and collected saliva from a bunch of people who were diagnosed with celiac disease and collected the saliva from a bunch of people who didn't have celiac disease. And then he did his chemical test and he looked at the results, and the results were kind of a jumble. And so as soon as he showed me the data at the end, I started asking questions like, well, what time of day did you collect the samples? You produce different amounts of saliva in the morning versus in the afternoon versus at night. He didn't know. Right? I asked, what gender are the people from? Are they male or female? Maybe naturally males and females produce different amounts of the chemical in the saliva that he was looking for. He didn't know. How old were they? We produce different amounts of saliva as we grow, or as we get older. He didn't know. So there was an example where he got saliva from like 20 different people. It wasn't easy for him to go back and say like, hey, can you spit in this tube again for me so I can run all my tests, and then I'll write down what time of day, what gender, and how old you are, and all this stuff, right? So collecting that kind of data, and maybe he wouldn't end up using it. But it's really important to think about it before you start. Okay. Questions? All right, I'm gonna go over hypotheses quickly because I know you've learned about them before, but I think I define a hypothesis a little bit differently than probably how you've learned it before. A lot of times when I ask for a definition, I hear it's an educated guess. Right? And that's true, it is. But I like thinking about it a little bit differently. I think of a hypothesis as one possible solution to a problem or a question. All right, so one possible answer or solution to a problem or question. There's usually more than one possible answer to any question, right? Yeah. So when you do an experiment and you write your hypothesis, there's never one hypothesis. There are lots of hypotheses. But you need to have a hypothesis written before you start. Science is most useful when you make a prediction. Think about this. Do you guys remember the eclipse last summer? There was a solar eclipse. Did any of you get to watch it? A couple of you? Yeah, so that, I mean, it wasn't great here. It wasn't like a full solar eclipse or anything like that, but you know, seeing the shadow go across the sun was pretty cool. But that eclipse was predicted over 100 years ago to the day, the hour, the minute. He was within a few seconds of predicting when exactly the full eclipse was, right? 100 years ago. Some guy predicted when the sun and the earth and the moon were going to be directly aligned with one another. That is crazy to me, right? But the important point I'm trying to make here is predictions are what makes science powerful. By predicting accurately, it shows that you actually understand what's going on. So like with the hormones for the plants, when you do your experiment, you probably are going to have a fairly good idea of what's actually going to happen at the end. You're going to have a prediction for what's going to happen with that experiment. Or you're going to be able to possibly answer how our hormones going to affect the plant growth. So again, I hated when my teacher would always ask me, what's your hypothesis? What's your hypothesis? Right? It's important. You can't do an experiment without having a prediction, without having some kind of idea of what's going to happen first. Okay? All right. I'm going to skip all this other stuff. You didn't know there were so many slides about hypotheses, did you? Okay. All right. Last two slides. Here are the most common problems that I think happen with doing experiments in middle school and high school. Okay, so this is kind of like the greatest hits of bad things. No clear question or statement. Right? You have to know what you're researching before you start researching. So you need a really clear question. These two kind of go together. Not collecting the right data or not collecting enough data. Like if we would, again, kind of picking on you guys, if it's like 3D printed stuff. When you have, let's say you 3D print 5 of the exact same object with different kinds of plastic. what, what are the things that you can measure? How strong they are, how light they are, whether they float or sink, their density, things like that. So collecting the right amount of data, ignoring important extraneous variables, not controlling your experiments enough.
John, can I add into that, looking into little data? We had a young lady last year in 8th grade who had her idea in October, waited until second or third week in January to try to do her experiment. Ended up, I forget what the issue was, but for some reason she ended up doing a 24-hour data collection, and that was it. When her experiment should have been, and it was with bacteria, and her experiment should have been at least 2 weeks long. All the teachers were, telling her. And so she ended up with putting all the work together to try to put together a science fair project, and basically had very little data to the point of where it was like she didn't even really need to take herself to science fair because she couldn't justify any of her comments, and kind of really had no idea how to come up with a conclusion because she didn't have the work there. As opposed to we had students who worked for 3.5 months over here once, twice a week doing work in the lab to get their project set up. So.
Yeah, and like this bullet point kind of goes along with that, like giving yourself enough time that you can repeat the experiment enough that you get worthwhile results. Like we had a great project a couple years ago with fruit flies and concussions. A girl took fruit flies, rattled them around in a tube, basically giving them concussions, and then stained their brains to see if a protein was produced. She was interested in concussions and Alzheimer's and things like that. She did it with 5 fruit flies in each group. And so even though it was a really cool experiment, the question is, if you can prove that or if you show something happening in 5 fruit flies, first of all, can you assume that it's true for all fruit flies? Is 5 enough to say, yes, this phenomenon is true of all fruit flies? Probably not. And second of all, if you do it with 5 fruit flies, can you say, this is probably the same in humans? No way, right? So we need to give ourselves enough time and repeat our experiments enough that we can be confident in our results. Now, if you do this with 1,000 fruit flies, and you get the same result pretty much every time, now you're feeling a little bit better. But what's the right number of trials, or the right number of patients? There's no magic number, but the more data that you collect, the better your results are going to be. Okay. Let me talk about this one real quick. Losing sight of the significance. Who cares about your project? Not to be too blunt, but like, did you pick a topic that's really significant? So I gave you the example earlier of imagine the most basic experiment you can possibly imagine, plants with and without fertilizer. We've done that before. We know fertilizers are going to help plant grow. All right, we can look, if you can Google it online and find an answer in 10 minutes, it's probably not a question it's not worth you doing that research again. So that's difficult at, especially in the middle school, and it maybe gets a little bit easier as you learn more and more as you go through high school and college and grad school, and then, you're people that are getting their PhDs. What is the question worth asking? The whole point of science is to learn something that no other person has learned before. And so you're not going to get there unless you know the right questions to ask. So losing sight of the big picture, why does this matter? And it's not like every experiment needs to be groundbreaking, especially not for you guys. But that should at least be in the back of your mind. Is this experiment that I'm doing now in my 7th grade year gonna get me to answering a really, really interesting and useful question? Okay. So if I can just leave you with a few things before you start your experiments, I'd like you to ask yourselves these questions before you start your experiment. What is your research question, or what are your research questions? What are you trying to answer? What data are you going to need to collect in order to answer that question? Can you physically do that? Some experiments, like the cell culture ones, might require some fancy equipment that we have here. Maybe we don't have them here. Bryn was just sitting over, Bryn was one of the interns sitting over there. She just left, but we had to go to Kutztown University to get a piece of equipment that allowed her to collect data. So thinking about that, do we have the equipment here? Do you have the equipment at home? Those are important things to think about right now. How do you measure the data accurately? What are the variables acting on your experiment? How can you control them? Right? Really important. And then finally, I think you guys talked about this a little bit more. Even though it's early in the process, you should already be thinking about, once I get results, how am I going to share that? How am I going to show it? Whether on a poster, how am I going to write about it in a report? Sharing what you find is super, super important. Science doesn't matter if you don't share the results with anybody else.
And one of the things we learned last year from talking to a couple judges after science fair was we discussed having our middle school have the same kind of display board as our high school students. And the judges I talked to all said they suggested not, because our high school students' displays are very professional, and it's almost now where people walk in and know they're Weiser students. So they suggested for middle school, just keep it where you're not necessarily doing it in a professional template, but instead we'll give you things that should be on there and then you sit and lay them out. And that's something that myself, Mr. Rasky, Mrs. Snyder, we will all sit and work with our students on when it comes time to actually put your display together. And some people do not need help on their display, they're phenomenal. And other people, 2 years ago, a young lady did a phenomenal experiment. She had the data she needed, and her strength was not displaying her information. She brought it in, and all her friends were like, we would have helped you. I mean, I heard so many students say to her, we would have helped you put that together. And she was going away and didn't have time to redo her display board, so she did not do well. And she didn't do well as far as being able to get her information across, except for to explain it to people, because when you look at her display board, it was pretty much of a mishmash mess. It really was, and it way too much white space. But that's something that, if you're one of the people that has a hard time visually laying things out, talk to the teachers, talk to your friends. I guarantee you probably have a friend that's phenomenal at it, and you may not even realize it, because every year when we look at these, some people are like, and just blown away by what people come up with, the ideas that they come up with.